I'm excited to have this conversation. I'm Tyler Smith, founder of 100. And look, it's really great to have both of you here. It's funny, before getting even into longevity, I used to follow and watch a lot of your videos, Ryan. It was interesting. I learned about peptides, I think, early on one of my videos. I think you were actually talking about SS31 or something. So maybe we dabble into that a little bit today, guys. So I'm really excited for that. Dr. Nick Norwitz, really excited to have you. Obviously, a lot of the recent topics you've been talking about have been really, really intriguing for me. I know we're going back and forth on a couple prescriptions, one I'm taking, so I'm curious to even dive into that realm. And so I'm really going to get my keto popcorn. That's right. That's for you, Nick Norwitz, right? I'm going to get my keto popcorn, sit back and watch this. I'm really, really excited. And before we get into things, I think just for the people who are watching, let's just give a quick intro. Ryan, if you want to start, give an intro on yourself, and then Nick, it'd be great, Dr. Nick Norwitz, to get an introduction on yourself as well, and then Ryan will kick us off with some questions.
Yeah, definitely. No, excited to be here with you both and dive into some of these new longevity topics. Just as a general background for me, I had really biochemistry undergrad, went to medical school, but just hated it. Ended up becoming an entrepreneur in several of the areas of science. My first company was tailor-made compounding, which, as you mentioned, Tyler, was a specialty compounding pharmacy that really brought a lot of the peptides to market that people are using today, including the SS31, which has recently got FDA approval, which is exciting and topical for the moment. But then, sort of after that, we were always looking at ways to validate that these therapies were working. And so got really excited about some of these biological aging clocks, particularly these epigenetic biological aging clocks, and created TruDiagnostic in 2020. And so in those last 5 years, we probably published over, I think, 80 different clinical trials now that we've done with DNA methylation, particularly looking in the fields of longevity. But really excited to talk with you both today, especially Nick. Been following your stuff, I think, ever since the Oreo experiment. So excited to hear a little bit more and go from there.
Yeah. Thanks. I'm very excited to be here, guys. Thanks for having me. I'll try to keep my intro a little bit short, but I think it sounds like I followed a similar path to you, Ryan. Interested in clinical medicine, found myself very frustrated with it, given a personal struggle I had. So by way of brief background, when I was going into grad school, so before I went to medical school, I just graduated up at Harvard Med. I did a PhD at Oxford. And as I was starting that, I was grappling with my own health problems, and the conventional approaches didn't really work for me. It's not that they're bad approaches, it's that they aren't built, and we can maybe get into this, to address the roots of chronic health conditions that plague most of society right now. So that's a whole different topic about the infrastructure of medicine. But that personal experience was sufficient to get me curious about the broad space I call metabolic health. Maybe using terminology, it might be a little bit more familiar to people, like deep biology and physiology. You really understand what is going on in every individual human and how we can use that knowledge, how we can leverage that knowledge to generate personalized medical protocols that can have profound impacts, unlike anything we see now today in modern medicine. And I think with evolving technology, we can get there. So we're going to talk about that. But that personal experience was the framework that kind of was behind me as I went into medical school. And I just kept on going through and seeing what I thought were dysfunctions in the system. So 5 months ago, I graduated, which meant I'm 29 now. I think I've spent 98% of my life like in school. I'm finally out of it and figuring out what I want to do with my life. And what I've decided is, look, there's so much amazing information. I want to be out there on the front lines, reading it, communicating it, and then collaborating with people to, one, advance the science, get awesome studies funded, but two, like turn this information into powerful tools for people to use so they can have these incredible individual transformation journeys. I think we fundamentally need a new way to look at medicine. And I think we're approaching that. Maybe we can get into a little bit of the details today, but even just by way of example, it's a little bit about me, tell my background a little bit different every time. So maybe that landed with someone, but excited to be here for this conversation.
Yeah, certainly. And I, Tyler, I think maybe just to jump right into it, I know we've got a couple of things we want to reflect on today and some of the new publications that have gone on lately. And so I know that we probably wanted to start with this one paper that was published in Cell Reports, which talked about sort of an individual analysis, but a multi-omic analysis of the woman with the longest confirmed lifespan, which was verified as the sort of oldest living person in January of 17th of 2023. when she was 117 years old. And so this multi-omic analysis sort of goes into sort of asking the question of maybe what is unique about this individual that sort of signifies longevity. And so I'm assuming that you both have had an opportunity to read this article.
Yeah, I think just taking a step back and saying what we can and can't conclude about this and really what the purpose is and why I'm excited about it. This isn't n equals 1. So, like, with all the caveats, we're not going to be able to draw extreme conclusions about, like, oh, this thing contributed to her longevity. That's not the point. The point is more, I guess, for me, about the methodology and hypothesis generation. You use this term multi-omic. So to unpack what that is, that means multi-omes. What are omes? Well, genome, all your genetic material, proteome, the composition of the proteins in your body, transcriptome, the transcript, etc. And so we're developing technology now, and we can give other examples about how this is now being used for research purposes and clinically, where it's not like you just go in and get a lab test, but you go in and you get these comprehensive analyses that give a high resolution picture of what you are as an organism. So it's kind of like the difference between drawing a sticky note sketch of a person. It's like, hey, here's your lab report, versus like something in 4K. So that's where we're going. That's actually now becoming more and more, available for research methodology, but even being turned into direct-to-consumer products. But that aside, this is super cool because you have the oldest living verified person. She was 117 and 168 days. And you get to look into like what makes her tick and then dissect out things that maybe fall in line with expected patterns and then things that don't, which is always the more interesting thing. And so it was amazing fodder for hypothesis generation. And I guess a few things I want to highlight that stood out were some of the markers that did make her unique. She had incredible mitochondrial health. So I think the term they used in the paper was not only preserved, but robust mitochondria. And I think that's very cool for two reasons. One, I mean, it sits with my confirmation bias that mitochondria are the root of like health and chronic disease, but also this is a very actionable thing to target. So even if you aren't blessed with the genetic lottery, we can kind of, capitalize on mitochondrial health to extend your health span, if not your lifespan. Other things that popped out were her epigenome, which I know is an area of expertise for you, which we can dig into, and then her microbiome, which is also incredibly youthful, versus other things that weren't unique about her. Like her telomere length was actually incredibly typical. She was like on a very normal population trajectory. And we think about that as usually very important. So her case helps at least start to dissect things that actually might be important and actionable from other things that we might have put more value in the past.
Yeah, definitely. That's where I was going to go next. Is speaking to where I have some particular expertise. To reiterate, I think a lot of things that Nick mentioned. This is just one trial, but I think it's super interesting. I mean, we all probably treat ourselves as N of ones, right? And always reflect on maybe how some of the patterns that we're seeing here might be things that we see within ourselves or even within the population that we service at TruDiagnostic. But I was a little disappointed actually in some of the clock analysis here, because as much as I'm excited about it, there was really only one second generation clock from an epigenetic perspective. And so I've already sent an email seeing if we can run some of the newer clocks on this, maybe even some of the organ specific clocks. But right now, the only sort of second generation clock that was run was Morgan Levine's Pheno-H clock. This has been a clock that is, really, exciting for whenever it came out in 2000, sort of the 17 area. This was the first clock trained on those blood-based biomarkers translated to epigenetics. And in the Levine clock, again, we're seeing ages of 106, 104 across the multiple different age samples or different types of tissues. But again, unfortunately, the only tissue that's been validated for the Pheno-H clock is blood, where we're seeing around 104. So I was a little disappointed in the epigenetics. Thought it could have been a little bit more robust with maybe some of the newer information, but still ultimately seeing all of the clocks, multi-tissue clocks, skin and blood clocks, all these first generation clocks be significantly younger, some as low as 89. The Hannum clock in particular got as low as 83 within urine, 89 within blood. And that's really exciting too, because it pairs well with some of the other data sets. So the Hannum clock is really based on a lot of these immune function markers, particularly incorporating the immune system. And so to see some other markers of the immune system, I think being rejuvenated or a lot younger, I think also fits with this picture of some of the big hitters, right? Mitochondria being super important, but also the immune system being pretty important. And so I think that was super insightful to see.
So we talked about this idea of multi-omics. Well, this can be done on lots of different people. And then combining it with machine learning, AI, these new tools we have, we can start to dissect patterns that no clinician could ever see. So I don't know if you know about the work of Michael Snyder. He's a researcher at Stanford, but his lab is all about multiomics. So they did a similar analysis, but in a different population of people. Now I'm going to talk about diabetes because I think it's something that pre-diabetes, diabetes is familiar to a lot of people. First, think back to that sticky note sketch versus 4K resolution differential. How do we diagnose diabetes, pre-diabetes today? You do something very rudimentary, which is you get an average blood sugar, or you do something maybe if you're a little bit more advanced, like an oral glucose tolerance test. So you take a bolus of sugar and see how your body responds to it. But even that's very rudimentary. So what they did, and I'll just kind of get to the punchline, is they're there's lots of different reasons people can have poor blood sugar control. So they did state-of-the-art lab testing to look at different reasons. So one was liver dysfunction, one was muscle insulin resistance, one was beta cell dysfunction, and one was incretin dysfunction. And then they put CGMs on people. So this is something you can get over the counter now, just get a CGM curve. And they used AI to look at the big data set and say, hey, is there something in the code, the exact pattern of the CGM response that actually gives an indication into what the underlying pathology is? And actually they found there was. So now they have algorithms where you can just wear a CGM, eat a potato, and they can say, hey, not only do you have glucose dysregulation, but we know specifically your flavor of insulin resistance glucose dysregulation is because of your liver or because of your muscles or this, that, and the other. And that level of precision, you were talking about the aging clocks in different organ systems, provides this incredible, sorry to use the word again, but precision by which we can address chronic disease, longevity, causes of death. Actually, one thing that speaking about aging clocks, the first author pointed out to me is a question on his mind is, why did she die? It's funny because this is a 117-year-old woman. We all kind of just say she died of old age, right? That's such a hand-wavy nonsense comment. It's something failed in her. She had an Achilles heel. And there's this underlying assumption that the body just conks out at some point. But that, our body's a machine. So if we can start to dissect using these multi-omic approaches, being the young people with chronic diseases or older people, what is your weak point and then target and strengthen it? There's no reason we can't extend the lifespan and health span, I think, potentially indefinitely. So it just got me so excited because of how we're looking at people as organisms now, which is totally different than any other era.
Right. I think that maybe some people might have been a little bit surprised to see that some of the HDL markers were really good, but the LDL markers not as much. So I love your thoughts on that and your reading of that.
Yeah. So just as a high level commentary, if people have seen the content I've already put out on this, I said, heavy quotes here, her LDL was high. Yeah. So what do I mean by that? Her LDL cholesterol, some people call it the bad cholesterol, was not astronomically high, but it was out of range. So the reason this is interesting to me is because I want to draw a contrast between what we're talking about here, this high resolution multi-omics, putting everything in context, seeing the individual as this complex being versus seeing things in isolation. And I think that contrast lies at the root of where medicine needs to go versus where medicine is. Everybody's had that experience. They go to their doctor's office, they get a blood test, the lab report comes back. The way the lab report is set up is there's reference ranges. And then if you're out of a reference range, the lab value is literally red. So it stands out. And that's seen in isolation and then treated in isolation. So it's very black and white, very algorithmic, out of reference range. Here's a lab marker, you smash it down. And then that brings up the question, well, why do we focus on particular markers? So LDL has been a focus. And I would argue it's a focus not because it's actually a good marker of risk, but because we know how to hit it with medications and lower it. So it's an easy win from a medical perspective. And that's not saying you should lower it. I'm just saying, or shouldn't lower it. It's just that we hit at the low-hanging fruit of what we can address because of the business model of medicine. There have been trials that have developed drugs that hit this biomarker, which can help in some people. A lot of people, it doesn't help. Actually, most people, it doesn't help. And we get into that a little bit later. But I just want to draw that contrast because I think people assume, quote unquote, evidence-based medicine refers to the best care. And what it actually refers to is, well, we have trials that have checked certain boxes, and these trials have been done because there's a business model and infrastructure behind to do those trials versus what actually matters. So in the case of this woman, to bring it back to this study, her LDL was high, but quote unquote high as in out of range, but that was completely de-emphasized. It was shoved over into supplemental figure 8B in this tiny line. And I don't think that's because the researchers were trying to hide it, like some conspiracy. I just think they thought, hey, in the full context of this woman's profile, overall, she had an excellent lipid profile. She had markers suggesting a very good overall, they used the term engaged lipid metabolism, so rapid lipid turnover, low, triglyceride-rich particles, et cetera, et cetera. So on balance, her profile was great. But that one marker that gets so vilified was out of range. And so what I conclude from this is not that she's a unicorn, not even that this marker doesn't matter, but that this is consistent, I think, with the broader data that this marker really doesn't matter that much for health span and longevity, but it takes up a disproportionate amount of airspace in the doctor's office and public conversations because of the tools we have, not because of its relative importance. What do you think?
Yeah. I think that it's all about relative importance, right? And I was even going to phrase this to Tyler as well. You know, with a company that measures so many biomarkers, it's oftentimes like, where do you start, right? How do you choose what is the biggest priority? So, Tyler, do you have any thoughts on that? How you typically tackle that?
Well, we attack it from all-cause mortality on how we give our protocols and how we think of meeting the patient where they're at. But we do well over 100 biomarkers, so it's a pretty comprehensive list. It's so funny. I was going to ask specifically for both of you guys on the longevity side. You mentioned LDL is not really tending to go towards longevity. That's what your hypothesis is. I'm curious, what would be the targeted markers that you'd be looking at? What would be the what items would you be targeting to the patient against specifically longevity needs? Because when you come on to 100, you get asked the question, what'd you come here for? High blood pressure, weight loss, longevity, preventative health, et cetera, et cetera. So I'm so curious on the longevity perspective, what specifically would you guys be thinking are the most important things specifically for that person coming for that reason to target?
I think generally an overall pattern that suggests good metabolic health. So as we saw in the super centenarian, low levels of inflammation. So they use in the paper like GLIC-A and GLIC-B. You can get an hsCRP, a cytokine panel. If all those are low, that's probably a very good sign. And then other general markers of metabolic health. So in a lipid panel, this will be the pattern of generally higher HDL and lower triglycerides will be a marker, and you can get more advanced lipid testing as well. Something with respect to insulin resistance, so maybe even a fasting insulin. It's kind of a poor man's proxy, but will be sufficient. And then if you want to get a little bit more advanced, and this is me teeing up Ryan to talk about what are the best clocks, but we do now have integrated tools to look at biological age. They're a little bit hard to really validate, but I think they're coming along. So I'm interested to hear what Ryan's thoughts are on the evolution of the generation of these clocks and their utility.
Yeah. For me, I'm a little biased here because, whenever we tried to create the best epigenetic clock, one of the first things we did was build massive omic data sets where we measured all those different omics that Nick was talking about earlier. We had full genome, we had transcriptome, but then we had really in-depth proteomics and metabolomics. And then we were able to actually rank some of them, right? We were able to say which have the best hazard ratios to predicting all-cause mortality. And that was super interesting because we got to see a lot of things we knew, right? And then a lot of things that were really new. And ultimately, I think that this was super exciting to look at. But at the end of the day, it was way overwhelming. And I think that this probably goes to an issue that Tyler has within, hundreds as a company, again, is knowing not just what's important, but what also is the most changeable or actionable. And so we had lists of, we ran over 8,500 different metabolites, and we saw some really cool things we talked earlier about mitochondria and just its relative importance. We saw a lot of mitochondrial function biomarkers be in that top 10 list, even supplements like ergothioneine, popping off, and not just all-cause mortality, but high hazard ratios for every disease. But ultimately, again, it's still hard to be actionable. And so I think that's why we really like the clocks, is because we're able to use AI to combine all of that information together. And unfortunately, to measure all of those different biomarkers from proteomics to metabolomics is prohibitively expensive. And you need to do things like collect plasma and, a lot of it in order to do that analysis. And so that's why, the sort of the clocks for us are a way to summarize that information all together, and then to validate it based on, how predictive you are of things like death, or when you can predict death. And it might sound really morbid, but, our omic age that we publish can predict death with a 92% accuracy within a 5-year period, which is pretty accurate. So we have a good surrogate way to measure death, but now we need to look at the clinical trials to figure out what changes that marker. And that's actually been really tough. And as just an aside on all of the different clocks, the mortality trend clocks are also the ones which are the hardest to change in our interventional trials, which might make sense to a lot of people because mortality is so difficult to change, right? It's hard to see those big increases to the same ages that this one woman was at 117, or any of the centenarians. But we now have these ways to track it in ways that maybe are a little bit easier to understand, instead of looking at 50 to 100 different blood markers. And so for me, I think that certainly the epigenetic clocks are certainly interesting to look at, but the newer clocks are able to integrate all of this omic information to not only be predictive but also actionable. And I think that's really where it becomes very difficult.
I am curious, have you seen any interventions, drugs or protocols that's moved the mortality clock at all, like in a real world trial or anything? I'd love to hear a little bit.
Yeah, the biggest effect size that we had across all the clocks was actually hyperbaric oxygen. And so two atmospheres of hyperbaric oxygen across the 3-week time period. And that was really interesting. And Luigi Ferici, who's the head of the National Institute of Aging, has also seen some interesting things, I think, in terms of hypoxia and some of these other conditions. And so that might be an area to really look at for interventional treatment. But with that being said, we've seen some really good things across the board with a lot of surprising things. But it all comes back to what we probably already know, which is diet and exercise are really the biggest movers across the board.
I would tend to agree. The only thing I'd add to that, so first of all, 100% agree. In terms of like action points, it's funny. It kind of comes full circle. Like you delve into the data, and it always comes back to, sleep, diet, exercise, introduction, just with more resolution. But you always come back to those things. So it sounds boring, but there's a reason it comes back to those things. That said, I do think that there are advancing technologies that could be very exciting. So Ryan, I don't know if you've seen any of the literature now on like small extracellular vesicles. And rejuvenation. So it's very cool. We start to think about what are the compounds floating around in our blood that can potentially be transferred or generated and transferred that confer youth, that rejuvenate our organs, that signals the mitochondria saying getting more efficient and younger. And there are like these little literal packages, these little spheres that carry information in the form of sometimes microRNAs, so very short little RNAs that change transcription patterns. And what they found is that you can transmit these between organisms and basically rejuvenate them. So they've done this in animals, so in mice, and have been able to restore the youthfulness profile. But the interesting thing about these pathways, whenever you look at them in different organ systems, is they appear very conserved. So the same information codes, packets, the microRNA, are conserved in humans. In fact, in the one experiment I'm thinking about, which is in Nature Aging, they actually did an experiment where they took the human, young or old, little packages and put them into mice. And they could still recapitulate the effect. And I was actually reading a paper today. This is tangential, but you'll like it. It came out, I think, last night in Cell Metabolism. And it was about transferring, again, probably through, it was through microRNA. I don't know if it was small extracellular vesicles, but the benefits of exercise, back to exercise, to your offspring through your sperm. And so what they showed that exercise changes microRNA that then gets transmitted through sperm to the next generation, so they could have mice exercise or not exercise and then test the muscular physiology, the fitness, and then the overall function of offspring mice. And the father's exercising was a transgenerational phenotype. And then to bring it back to the human, they're like, hey, let's identify what little packages are that are being transmitted. They found 10 in particular, and I think 7 of them were completely conserved in humans. Mm-hmm. The reason I bring this up is because I think it's very interesting to see this as conserved biology, and that we can start to isolate the factors in our biology that can be transferred. And these aren't complicated things either. So you could develop them basically ex vivo and think, I don't have any affiliation, but there's one company that's actually, it's called Mitrix. I know Michael Snyder's involved with it. I mentioned it earlier, which is ex vivo doing these mitochondrial bioreactors where you can just like build mitochondria potentially for transfusion into humans to rejuvenate them, because we actually have free mitochondria and mitochondria in these little vesicles circulating around in our body. Our organs use them to communicate. So they're not just like little stationary engines in a cell. They like travel around the body. And we're getting close to the point where we can just like generate them and put them into bodies. So 5-10 years out, I think that could actually be like a available therapeutic that people could be using quite safely, because it's not like you're changing your genetic code.
Yeah.
You're just giving your body more youthful engines, and your cells can take them up. So
yeah,
it's pretty cool.
Yeah, the mitochondrial transplant stuff is awesome. I think that even that recently they had the first ever official human mitochondrial transplant from the owner of, I think, one of the mitochondrial transplant companies. So yeah, it's super interesting, and that whole realm of research, starting with like the heterochronic parabiosis, right, where they sew up the two systems of a young and old mouse. It's super interesting, and trying to figure out what is the secret sauce, right? And actually, one of the interventional trials we looked at in that study was umbilical plasma, where they just took a umbilical plasma and injected it into older individuals, and we saw biological age reduction, which is super interesting. But we still don't, I'd say necessarily know what the factor is, right? What the magic sauce is there. But even we know the therapeutic plasma exchange now in 2 trials where we're taking out some of maybe the bad stuff and filtering it, and still had some of those super positive effects on at least some of our markers of biological age.
Like, how can somebody, a real world consumer, myself, take these things, understand them, think about them, and go, okay, how can I start to put these in practice? Or what is there to put into practice? Or what should I take away from this? That's what I'm always trying to think about. It's like, this is really, really insightful and really an exciting time for all of us, I think, to be alive. But like, when do these types of therapies or things come available to everyone, right? Or is it just to the uber ultra wealthy? When does it come down to a reasonable price? What action can folks take? You have everyone just saying, stay alive, all this stuff is coming. But when is it actually here? And when can people start to consume it? And so that's a big question I always ask, because look, this is all super fascinating. But I think a lot of people are like, how do I put this into action? Or what are some things I should be thinking about today, right? Not 5 years from now when this is not on a mouse study, or, the plasma is not hype, it's actual fact, and there's trials. And I know those things take a long time. And obviously with a lot of changes going on, we're seeing those a lot faster, but still, how can the average day person really take this, understand it, but then put it into practice? Or what should they put into practice?
Right now, this isn't mass medicine, right? It's still, small trials on study subsets that are maybe not the most indicative of the population, and so, I'm not sure how much of what we're talking about or getting excited about is, ready for prime time. But I think there are certain things that are certainly ready for prime time, and we always start with, the diet, exercise, and nutrition. But I think that some of those things that are working on pathways we already know are super important. Just to bring this full circle, we did start off the conversation with SS31, a product which increases and improves mitochondrial efficacy, and now FDA approved under orphan drug approval. I think that some of those things are hitting pathways that are tried and true, things that we know improve mitochondrial function, which helps every cell in the body. I think that those are usually where my picks go, or to things that are pathways we know that are tried and true and really efficacious.
So kind of 2 answers. We can talk about precision supplements, things like, creatine, vitamin D. I think omega-3s, if you don't get a lot of fatty fish, are probably good for everybody at some level, especially if you are predisposed to, say, Alzheimer's, have an ApoE4. And then talking about the fundamentals and how nerding out, which I'm guessing if people are listening to this point, they're probably already kind of on nerding out level, how that can serve as a motivating factor. I mean, let's be honest, nobody's listening at this point who isn't a little bit sold. So that's kind of how I think about it at a high level. But thanks for helping us try to ground that in actionability.
Yeah. And maybe, to take this, separately, I know one of the papers that you had mentioned was sort of about the mitochondrial transfer and how that sort of was affected in obesity. But I think we've talked about a lot of, good things, right? The things we want to do as interventions, but let's also talk maybe about some of the bad things we want to avoid, right? I think that, that paper was super interesting. I think there's some other papers which, talk about statins even maybe being, not as good for the mitochondria. So Nick, I think first and foremost, maybe a question for you is, let's talk about some bad things to avoid.
Yeah. All right, let's make mitochondria the theme. I'll start off with the controversial one, which is statins. And the reason I aim at this, I want to be clear, is not because I think statins are rat poison or that people shouldn't take them. But I think in any evolved discussion, be it supplementation or prescription medications or anything else, it's always an individualized cost-benefit analysis. And in order to really do that, you need to truly understand the cost and the benefit, or alternatively, understand what the unknowns are. So, one bit of coverage I did recently was on a human trial that showed the most common statin, atorvastatin, which goes by Lipitor, harms mitochondria. In fact, in asymptomatic humans, even at nanomolar doses, inhibits complex 4 of mitochondria by about 50%, with effects on muscle physiology. Now, the reason I think this is important is, one, not everybody will benefit from a statin, and in fact, a lot of people will be harmed. And trying to figure out, what camp you're in requires a little bit of, work. Are you someone who has had a heart attack or has pre-existing severe cardiovascular disease? That's very different than somebody who is, otherwise entirely healthy with one marker out of range, like this 117-year-old, right? In which case this intervention could actually be potentially harmful.